Welcome to the third video of Chapter 3, Nucleic Acids and the Encoding of Biological Information. In this video, we will discuss the mechanism of transcription in the cell. Transcription begins when the promoter region of a gene is recognized by factors in the cell called transcription factors. Transcription factors are proteins that recognize the specific spelling of the DNA sequence that makes up the promoter and bind to that region of the DNA. In eukaryotic cells, there are at least 6 different general transcription factors that must bind to the promoter before transcription starts. In a prokaryotic cell, like bacteria, there is only one protein that binds to the promoter region, called the sigma factor. Additionally, there are other specialty transcription factors that are called transcriptional activator proteins, which bind to regions upstream of the DNA called enhancer sequences. Not all genes require these specialized transcriptional activators. Once transcription factors are bound to the promoter, they recruit an enzyme called RNA polymerase, which binds to the transcription factors and interacts with the DNA. There are different types of RNA polymerases in the cell, but the main one, which transcribes protein encoding genes, is RNA polymerase II. RNA polymerase will unwind the DNA double helix and build the new strand of RNA by covalently attaching the ribonucleotide building blocks. RNA polymerase functions by first ensuring that an incoming ribonucleotide is the correct base pair match to the next nucleotide in line on the DNA template strand. It ensures that the ribonucleotide is oriented correctly with its 5' phosphate facing towards the 3' hydroxyl of the RNA strand. The next step is that the 3' hydroxyl of the RNA strand then attacks the high-energy phosphate bond of the next nucleotide to be added to the chain, forming the phosphodiester bond that connects one nucleotide to the next. This process will continue as RNA polymerase reads the template strand of DNA until it reaches the terminator sequence and transcription stops. The RNA transcript produced by RNA polymerase is known as the primary transcript, and it contains the information of the gene that was transcribed. In prokaryotic cells, the primary transcript is the final product and is ready to be translated into protein. RNA that will be translated into protein is called messenger RNA, also known as mRNA. Thus, in prokaryotes, the primary transcript is the same molecule as the messenger RNA. In fact, because transcription and translation both occur in the cytoplasm of a prokaryotic cell, they can actually occur almost simultaneously. Translation does not have to wait for transcription to end, as seen in this figure. Unlike prokaryotes, in eukaryotes there is a barrier between the process of transcription, which occurs in the nucleus, and translation, which occurs in the cytoplasm. Because of this barrier, the primary transcript in eukaryotes must undergo a complex process of chemical modifications known as RNA processing. This piece of DNA has been transcribed by RNA polymerase to generate the primary transcript. The first step of RNA processing is that the 5' end of the primary transcript is modified by adding a special nucleotide called the 5' cap. Next, the 3' end of the transcript is modified by the addition of a poly-A tail, a long string of adenine nucleotides. The 5' cap and poly-A tail allow the RNA to leave the nucleus and protect it from being degraded so it can be translated. The last step of RNA processing is called splicing. In eukaryotic genes, there are regions of the DNA called exons and regions called introns. In the figure here, the exons are in the dark color and the introns are in the pale color. The exons and introns have corresponding regions in the primary transcript. The genetic code is stored in exons only. Thus, the introns need to be removed before RNA is translated. The mechanism to remove intron sequences from the primary transcript is called splicing. In splicing, the RNA is cut so that one exon can be joined to the other exon and the intron removed. After this process is complete, the messenger RNA is now ready to be translated. One way to remember the splicing step is to remember that introns are in the way. Thus, they must be removed before mature messenger RNA is produced. Splicing also provides eukaryotic cells a mechanism for producing different products from the same primary transcript. A process called alternative splicing allows the exons of some genes to be rearranged in different ways during splicing to produce different mRNA products. If different mRNAs are made, ultimately different protein products will be produced during translation. This process is key for producing all of the antibody proteins that are a key part of your immune system's defense against invading pathogens.